Everyone, welcome to this video, and in today's video we'll be discussing about the hemoglobin buffer system. So in last few classes we discussed about the acidosis and the alkalosis and have already made the diagram for you. So this is the tissue and this is the blood vessel, right? So how our blood also act as a buffer system, so that we are going to study in this particular lecture, right? So in tissue, so oxygen respiration cause the formation of the carbon dioxide, right? So now this carbon dioxide will be distributed towards the blood so that it get recycled in the lungs, right? So 10% of the carbon dioxide get dissolved in the plasma, okay, and the rest carbon dioxide get attached with the hemoglobin and they form carbaminohemoglobin, okay. So they form carbaminohemoglobin and that is around 20%. So 30% of the carbon dioxide is recycled in the either in the form of the carbaminohemoglobin or the plasma, right? And the rest is same as that we discussed. So the carbon dioxide first goes into the red blood cell, then what the carbon dioxide and water will form H2CO3. The H2CO3 is going to divide into the proton as well as bicarbonate ion. Now what happens is, so bicarbonate ion get out of the cell and there's a chloride shift that occur and it form HCl inside the system, okay. And this HCl get recycled in kidney. It's as simple as that. You do not need to remember much, but this is the whole scenario of the hemoglobin buffer system, okay. So here what is happening is, so there is a transport of oxygen from lungs to peripheral tissues, my bad, and the carbon dioxide is transported from tissues to lungs, right? Now hemoglobin is buffer for both H plus and CO2 is buffer, okay, H plus and CO2. So carbon dioxide diffuses across the RBCs. So like I discussed, so this is RBC. So carbon dioxide can easily diffuse and it can merge with the water inside and it can form H2CO3, okay. And carbon dioxide can also bind with plasma. So like direct dissolving of the carbon dioxide plus water, right, with plasma. And carbon dioxide can also attach to hemoglobin to form carbaminohemoglobin, okay. So this percentage is around 70% and the rest of the two are 30%, okay, that you need to remember. Now coming to the phosphoric acid species, right? Phosphoric acid species. See guys, these topics are very short topics, but if you understand them once, you'll be able to perform any question that might be asked during your exams, okay. So phosphoric acid species are usually the conjugate pair. And what are the conjugate pair? So for example, we have H3PO4, right? So how many times this proton can dissociate? It can dissociate at 3 levels, okay. So if one H is removed, right, so it can form H2PO4 negative. Another H is removed, the proton is removed, it can form HPO4 negative, right. And if another H is removed, so it can form the phosphate entity itself, okay. So that is how the phosphoric acid species work. So because it has loads of proton, thus it can create lot of conjugate acid. So what is the conjugate acid? If you do not know, so watch the lecture 7, you'll get the idea, okay. So it can create a lot of this conjugate bases, and thus it creates the system where it can releases the proton when needed and it can create the conjugate base for the buffering action, for the buffering action. So let's just suppose if your system has much more proton increase, so what happens is, so either of this HPO4 negative, H2PO4 negative can come, or even the PO4 negative can come and attach to this proton and they can form this, either the H2PO4 negative moiety or the H3PO4 negative moiety or HPO4 negative moiety, okay. Any of these can be formed. And if there is much of this OH negative ion, so what a phosphoric acid will do, so it can dissociate its proton and it can form the water as well as the PO4 negative species. I hope you get the basic idea on how a buffer works. If not, please watch the lecture 5 till lecture 9, and these are like 5 and 6 minute videos, you'll get the idea. So now we'll discuss about the proteins. So now we are coming to the biomolecules. So first we are going to learn about the proteins and how it works. So first we'll learn about the amino acids. So what are exactly amino acids? So these are building blocks for protein. Okay. So if we look at the very basic structure, so there is a alpha carbon. To this there is a carboxylic acid group. There is a NH or the amino terminal. There is a hydrogen species attached to it, and then there is a R species which differs the protein structure. Okay, and this R can vary from like one hydrogen to many different groups. It can either it can have a hydrophilic group or the hydrophobic groups or simple hydrogen. Okay, due to which what happens is, so there is a chirality that occurs. Now what is chirality? So for example, we have a amino terminal, that is NH2. To this there is a alpha carbon. So the mid carbon, so from where all of the attachments are happening, is called alpha carbon, and the group, right, R is basically differs for every amino acid. So then there is a carboxylic acid group, and then there's a proton group, right? So now when you create a mirror image of it, so these images do not overlap. Okay, so अगर आप इसको उठा करके इसके सामने ले आओगे, so these are not going to overlap. Thus there is a chirality in amino acid structure. Okay, so what are the properties of amino acids? So first we have 20 amino acids, 20 amino acids. So I'm writing AA. So these are white, crystalline, high melting solid at room temperature. Okay, all amino acids are building block of protein that we just discussed. Amino acid also act as enzymes, as catalyst, metabolic intermediate. They are carriers of energy and waste product. Also in some cases they act as hormone and transmitters such as neurotransmitter, etc. Okay, so what happens is, so this I'm going to discuss further also. So for example, if we have 1 gram of protein, okay, let it be any kind of protein, so when it metabolizes, it gives us 4 kilocalorie of energy. Okay, so thus they are also a carrier of energy, and this happens only in absence of carbohydrates and fats. So when you are fasting and your body has utilized all the carbs, then the fats, then only it comes and break down the protein to convert it for energy. Okay, so this is the scenario. First carbohydrate will be utilized, then the fats, and then the proteins. Okay, so there is only one way to link amino acids to form a peptide bond. So most of you know the chemistry, I know, right? So for example, if I have NH2, C, COH, let's suppose H, and let's suppose there is CH3, right? So if any group comes, so for example, if another amino acid is there, so if these two amino acid has to bond, so they can only form bond in one way, and that is through this particular bond. Okay, only when CO of one particular amino acid and NH of the another group are going to attach, so this bond will be called peptide bond. Okay, so otherwise in chemistry what can happen is, there can be multiple different bonds that can be formed. So for example, so this is dipeptide. Okay, this is dipeptide. For example, what other thing that can happen is, so let's suppose CH2 and HCCH2 and this COH can also form, meaning this particular part and this particular part can form a bond. So this bond won't be called a peptide bond. Only where the CO and H bond is there, only that particular bond is called peptide bond. Remember that this question comes in both the GAT B, GATE as well as ID exam. That what is a peptide bond, and they'll give you like multiple different things. So for example, they'll give you NH2 COO, so this is a peptide bond, or NHCO, is this a peptide bond? Yes, this is the peptide bond. Okay, so they can write in a very different way, but you have to use your mind and you have to choose the very correct option. Okay, so that is why we need to understand what exactly is the peptide bond. So this bond is peptide bond, just remember that. So when carboxylic acid of one end and the amino portion of another end form, and in this formation they exclude the water molecule. Also remember that in this particular bond formation there is a loss of the water molecule, because COOH is losing the OH part, right? And NH2 is losing the hydrogen part. So thus there is a loss of water. I hope you get it, and if you did, so write amino acid in the comment section and I'll know. So I'll make the next video about the different type of amino acid that we have. Thank you.